Excellent. Let me get my pointer. So today is lecture 15. We will be covering sensory systems of fishes. And we're going to talk about how different fishes can sense different things and what their capabilities and limitations thereof are. So lecture 15 in your syllabus. So I'd like to just begin with an overview here of sensory systems in vertebrates. And the fishes have the same 5 senses as other vertebrates, you know, sight, smell, taste, hearing, and touch. And so these are the actual physiological terms here for this. You know, hearing is hearing, touch is touch. But sight, typically we call vision. Smell is called olfaction. And taste is gustation. To gustate is to taste. And so, like other vertebrates, fishes have these senses. Now, we've already talked about certain fishes, for instance, that live in the deep sea or that live in caves away from light. They are blind. And so they might lack vision. And so not all fish possess all 5 of these senses. You know, some fish might elect to lose a sense. Alternatively, we talked about how some fishes, Ostariophysi, which includes, you know, things like family Cyprinidae, the minnows, have very good sense of hearing. And so in some cases, these senses might be more sophisticated in one group of fish or the other. The take-home message here when we talk about senses is that they're not free, okay? And from free, I mean, we ended last lecture talking about bioenergetics and how energy breakdown occurs inside of the organism. And for instance, there's a cost that comes with making eyes and running those eyes. You know, it takes energy to power an eyeball. And if you don't need it, why are you wasting that energy? You could be dedicating it to something else. And so all the senses, there's a trade-off. So some senses might be very important, other ones might not be so important. And sort of that has to do with the life history of the fish, its environment, what its ecological niche is, all sorts of things. So there's a trade-off there with the senses. But generally speaking, all fish have the, I guess, capability or possibility of having each of these 5 senses. Now, there are 2 additional ones I list here. That is the lateral line, which is acoustico lateralis in Latin. This is distant touch. And so imagine if we took the sense of hearing and combined it with touch. That is sort of what this sense is, acoustico lateralis. And then also we've already talked about how some fishes can sense electricity. There is also electrical reception. And so fishes have these basic 5 vertebrate senses in addition then these 2 senses here. So I would say that fishes have 2 or 7 total senses, and really there might be an 8th in there that we'll talk about later. It's electromagnetic sensations, which we don't really know a lot about, but we can sort of see that fish know how to do it. So there might be an 8th sense in there. So when we talk about what these senses are, sight is very similar to terrestrial vertebrates. However, a major thing to consider here is exactly what I said in the very first lecture, is that water is a lot different medium than air that's around us. The refractive index of water and therefore the lens of the eye is different. And this is because water is more dense than air, and therefore it bends light differently when light enters it. Also, there is less light at depth, and we talked about this before. Red wavelengths attenuate in shallow water, meaning that there's no red wavelengths of the visible spectrum getting into the bottom of the ocean. So there's no need to see the color red because there's no light down there to bounce off an object to see. So color vision also then is less important for some species, especially those that live in the deep or in caves away from light. Hearing is more difficult for fishes compared to us terrestrial animals. And that is because if you consider an organism as a bag of water, you're mostly water that's contained in some kind of integument. And if you're a bag of water living in water, that means your density is very similar to that of the density around you. And therefore, you are largely transparent to sound waves. That means that in order for you to perceive a sound, the sound really has to travel from one medium into another, meaning that it has to change density. And so from going from air to a physical thing, a structure like your ear, that's two different densities. Air is a lot less dense than your eardrum. And so the fact that fish have a very similar density to what's around them means that hearing is actually kind of a difficult sense. Part of the reason why this acoustical lateralis system is pretty important, because it's a way of hearing in a slightly different way. And I also then mentioned some fishes that have adaptations to overcome this. Ostariophysi. They use the swim bladder, which is filled with gas, to transmit the sound. So a sound wave goes from water into the fish, both of which are primarily made of water, same density, but then they bounce off that swim bladder, which is full of gas. Now, gas is a lot less dense than water. So that's how then we can transmit that signal. We can sense it. So you can sense something when, you can sense a sound wave when it goes from one media to the other. Touch is virtually the same in other animals as it is in fishes. If you touch a fish, they can feel that. That's just a simple mechanical receptor. Smell and taste are very similar senses in fish. So we have a nose that contains olfactory receptors, and we have a mouth that contains taste receptors. But fish are essentially in the water, and anything dissolved in the water you can taste. And so they're essentially tasting everything around them all the time. And likewise, they can have taste receptors located outside of their body. So really the only difference here between taste and smell is which part of the brain processes the information. We have our taste receptors relegated to our mouth. Fishes can have them all over their body, not just in the mouth. So the way we look at it is where are the receptors located on the fish and how are they processed in the brain? Many species have taste receptors on their fins, on barbels, things like that. So not just in the mouth. So also, when we look at the sense of olfaction and gustation, generally, we look at olfaction being a volatile substance that's transmitted through the air, whereas taste is something we sort of put in our mouth and then evaluate that. And again, because fishes are swimming around in an aquatic medium all the time, everything is dissolved in water, and so they can process both olfaction and taste simultaneously just by swimming in the water. So they don't have to worry about physically touching anything or putting something in their mouth. So those are the senses of fishes, and then also an overview there of the vertebrates. So when we look at the eye of a fish, most fishes have binocular vision. Bi is two, so they have two eyes, one on each side of the head. Sometimes they're laterally placed, like here on the side of the head. Sometimes they're on the top of the head. And even in the case of the flounder, the flatfishes, they start off with eyes on both sides of the body, but then one eye migrates on the other side. They still keep that eye. So it sort of tells you that binocular vision is important, and this is because binocular vision allows you to orient three-dimensional space. If you have two eyes, it can sort of tell you a distance or proximity away because you're collecting data from two different sources. Because of the eye placement, typically on the sides of most fish heads, they have a pretty considerable blind spot in front of their head. So it's kind of like you touching the tip of your nose and trying to see your finger. You've got a pretty small blind spot. If you pull that finger out just like a centimeter or so, you can see your finger. So you've got a little blind spot in front of your head, but not as bad as a fish. And that's because of the lateral position of the eyes. So this blind spot is pretty considerable. And when you consider the fact that the fish needs to use its mouth, which is usually located right here, this is kind of a problem. But the bonus is having an eye on the side of your head means you have good lateral vision. A fish can typically see almost all the way down to its tail, whereas you cannot see behind your back. a fish actually can see behind itself. So the lateral eye placement, again, when you consider these things are living in a three-dimensional environment, they got to worry about a predator coming from behind, from above, from below, from in front, from the side. They have to be able to have pretty good senses here. But this blind spot in the front of the head is considerable, and we'll talk about the ways fish get around that. It has to do with the lateral line system. The eye looks like this, typical of other vertebrate eyes. There is a pupil here that light enters. There's a cornea that sort of protects the eye and keeps all the fluids and things inside of it. And then there's this lens. Now, a major difference between the lens of a fish and a vertebrate such as yourself is the eye lens of a fish is a perfect sphere. It has the highest refractive index. The lens of a human eye is not a perfect sphere. It's kind of oddly shaped. And when you go to focus your eyes, there's a series of muscles and ligaments here that sort of stretch or compress the lens. And that allows light to come in through the lens and then focus onto the retina, which is what senses the light coming in. Fish do not stretch the lens. This sphere is not malleable. What it does is it uses this muscle called the retractor lentis muscle, and this thing can contract or relax, and it literally physically moves the position of the spherical lens in relation to the retina. So it moves it either forward or backward or up or down. And that when that lens moves, then as light moves through it, it then changes that refractive index so it can focus on different parts of the retina. So major functional difference there between a terrestrial vertebrate and a fish eye. Some species that have a pseudobranch, and I showed this last lecture, that little tiny miniature gill arch underneath the operculum, supplies blood directly to this, what we call the choroid, or the choroid gland. So if you notice here, the retina sort of is in the back of the eye, and here's the optic nerve. Retina receives information and sends it to the brain through the optic nerve. The choroid sort of sits here right behind the retina and supplies oxygenated blood to the retina. Now, this structure, this retina, takes a lot of energy to run. Very expensive sense. By far the most expensive of the senses is a camera eye. And so light comes in through the lens, focuses on the retina, retina receives information, sends it down to the brain through the optic nerve, and the choroid is what supplies oxygen then to the retina to run this. It's the energy to run this. And so some fishes with a very high visual acuity or very good sight, predators for instance, have a pseudobranch and a very good choroid to supply oxygenated blood to the retina. And then the retractor lentis muscle then is what focuses the lens of the eye. This thing doesn't stretch, it literally moves position three-dimensionally. So that's a general overview of the fish eyes. Some specializations in fishes, for instance, let's look at elasmobranchs. Remember the strap-gilled chondrichthyan fishes, or chondrichthyan fishes. Elasmobranchs have very good vision. The retina has both rods and cones, sorry, which I forgot to go over that. Rods are for black and white vision, low light conditions. Cones are what give us color vision, but they only work in very high light conditions. So typically only fishes that are towards the surface. Deep sea fishes are seeing more like this. Surface fishes see more with cones. And nocturnal fishes also will use rods. So the retina of chondrichthyan fishes have both rods and cones, but it's dominated by rods. So these things see very well at night, and a lot of them are nocturnal predators. Some cool things here is that they actually have an eyelid called the nictitating membrane, or the nictitans. And see this flap here? It sort of opens from the bottom up. And so the sharks can close their eyes by pulling this membrane up. This eye is partially closed. It's covered by the nictitans. And this is because when sharks feed, it's pretty violent. They literally are just thrusting their head at something and ripping it apart with their mouth. and that leaves like chunks of debris floating in the water. The other fish is thrashing, you know, and it could damage the eyes. And so when they bite, they oftentimes pull this nictitans up so the eye doesn't get damaged during the feeding time. It's common in certain sharks like the great white sharks, blue sharks, tiger sharks, lemon sharks. Again, apex predators who are using this to bite other stuff so that their eyes don't get damaged. But other than that, their eye works just like the typical bony fish eye I showed before. And so again, the nictitans protects the eye from damage during feeding, which is a violent activity. Now here is another unique adaptation of fish eyes. I'm going to show you this. This here is kind of crazy. So look at this here. This is a barrel-eyed fish. Now, do you think that these are the eyes right here? If you do, you must reconsider. So watch this.
Photographed 2,000 feet deep in the Pacific Ocean off the California coast, the Macropinna microstoma, known as the barrel-eye fish, is small and dark with large fins, a tiny mouth, and unusual barrel eyes under a transparent dome. The two green spheres in the video are the lenses of its tubular eyes. The eyes are enclosed within the transparent shield, sort of like the glass canopy of a jet fighter. Above the mouth, the two dark capsules that appear to be eyes actually contain the fish's olfactory organs, or the equivalent of nostrils. Typically, the barrel eye sits quietly in the water, using its big fins for stability while it scans the water above for food. When it spots food, it can rotate its eyes to look forward, to include its mouth in the field of view. Scientists speculate that the barrel eye steals food from siphonophores, elongated jellies with tentacles that capture prey that swims into them. They believe the barrel eye swims into the tentacles and steals the food from the siphonophore. The shield over its eyes protects them from the stinging cells of the siphonophore's tentacles. The barrel eye can also rotate its eyes to avoid predators, or avoid being captured by scientists. The fish discovered alive by the Monterey Bay Aquarium Research Institute is the first specimen of its kind to be found with its soft, transparent dome intact. It had been known since 1939, but only from mangled specimens dragged to the surface by nets.
Pretty ridiculous. And so lots of adaptations there. As you can see, the eyes of the barrel-eyed fish are enclosed. And again, it's part of life history. If it's going to be feeding by taking away food items from stinging jellyfish, it needs to protect its eyes. And so lots of variation here in some of the eyes. But again, if you look at the biology of the fish, it sort of tells you sort of why the structures are the way they are. Also, adaptations, you know, a lot of terrestrial animals have this too. Anyone driving at night, when in the headlights on the side of the road, you can see a deer because their eyes are reflective. Very similar structures are in fishes as well. It's called the tapetum lucidum. And really what this is it's a reflective layer on the inside of the eyeball. And so if we go back to our figure here, light passes through the pupil into the lens. The lens then bends the light. That light then hits the retina. And some light can pass through the retina into the back of the eye. And what happens is the back of the eyeball, if it's coated with the tapetum lucidum, then reflects that light back out. And so that means the retina gets to take two pictures, if you will, or gets to see the data twice for every image. And so what this does is it enhances vision in low light conditions. So it allows light to bounce through the retina twice, once incoming and once outgoing. It also then allows you to see reflections of the fish eye. So here's the tapetum lucidum causing eye shine in a shark. This looks like it's a walleye, so family Percidae. This eye shine then is caused by the light literally reflecting right back out of the eye. So light comes in, retina takes a picture of it, bounces off the tapetum lucidum, through the retina again. Retina takes a second picture of it, and then out back to the eyeball and into yours, which is why you can see this. It also then aids the rods in detection during low light conditions. So a lot of times these are fish that are nocturnal, or they live in the deep, where light is a scarce commodity, if you will, for powering senses. And so here's an eye shine video showing some fish swimming by the camera, and you can sort of see their eyes reflecting the light from the camera. And whenever you see this as a trait or a feature, you can sort of tell a little bit that fish is either nocturnal or it sees very well in low light conditions. It's either deep water, nocturnal, something like that. And so, and again, you can see it in other terrestrial animals as well, such as deer are a good example. So that structure is kind of conserved among vertebrates. Tapetum lucidum causes the eyes shine. When we look at agnathans, again, the jawless fishes, they've got some adaptations to vision as well. The hagfish, remember from the video, these things, a lot of times, you know, the commentators on that video of the hagfish tumbling around on the bottom of the ocean in the muck, they said, oh, look at this eye spot here. This must be the eye. No, no, no. The eye is this tiny little discolored thing down there. This is just a part of the coloration of the body. This is not the eye. The eye is like this really tiny thing there. They don't have a lens. They don't have any muscles that control the eye. I would say they don't really have an eye. They have what is called a photosensitive patch that lies under the skin. And what this does is it allows them to, say, detect shadows. Like, is it light or is it dark? They can't really see maybe images, but they can detect whether or not light is present or absent. That's about the extent to their sense of sight. But again, when you think about it, these things are literally on the bottom of the ocean, tumbling around in the muck. And so they don't really need to have a whole lot going on there. And so we say that they're nearly blind. The lampreys have a very good camera eye, one on each side of the head. And the eyes can be very large in some species and very well developed. The sea lamprey, Petromyzon marinus, actually has photoreceptors located on the tail. Then they can sense light through this, in which case they avoid it. And so the eye is not necessarily the only structure of a fish that can be sensitive to light. Sometimes they have photoreceptive areas elsewhere on their body. And so some adaptations here of the agnathans. Moving on to olfaction, which is the sense of smell. Jawed fishes have paired olfactory chambers, each of which has what we call an incurrent and an excurrent nostril. Now, I sort of said generally in the beginning of the lecture and in the lab that most fish, when you see that they have a nose or nostrils, that these nostrils do not connect. They do not connect with the pharynx on the inside of the mouth. So the mouth opens, there's this pharynx or buccal chamber in here that connects to the esophagus, etc. The nares, unless it's a special case, which we'll talk about, this nares does not connect to the inside. Like, you can inhale through your nose or your mouth because your nasal passages of your olfactive sense connect to your pharynx. This is not the case in the vast majority of fishes. What happens is we have an incurrent side and an excurrent side, and sometimes there's a flap of skin here. Sometimes the nares is tubular, like in this moray eel. But anyhow, there are cilia in here that sort of beat. And when they beat, their wave-like action pulls water in the incurrent siphon down here to the olfactory epithelium, where the sense of smell is taken in. This is where the sense occurs. And then those cilia beat and the water then comes out the excurrent side. So it's what we call a blind sac. It's not attached to anything. And so The cilia then within those chambers move water into and out of the nostrils, which are usually small pits then separated by sometimes a flap of skin, maybe tubular here in the bowfin family Ameidae, also then the eels of Anguidae. Of course, this is a moray, so it's not an Anguid eel. In teleost, the olfactory chambers are dead-end sacs. They do not connect to the pharynx, except, you know, again, it's a special feature. Family Uranoscopidae, remember the stargazers, they actually do have a connection here between the nares and the pharynx. Generally speaking, it's rare. The nares of elasmobranchs, chondrichthyan fishes, are located ventrally on the snout, and they are also not connected to the pharynx. Chimeras, holocephali, the rat fishes, and the lungfishes, Ceratodontiformes, they have paired nares that connect to the oral cavity. So the chimeras and the lungfishes do. But again, lungfishes are kind of unique in the sense that they have a lung. So maybe they resemble the hardware here with smell is a little bit more closely resembling what we terrestrial animals have. So generally speaking, that is olfaction. This is typically a blind sac, and the sense of smell then occurs here. And then there's nerve fibers that relay then back to the brain. So a compound comes in here with the water, that compound is sensed, goes out, and then that sense is then relayed to the brain. Olfaction. Now, the inside of the nares is lined with a highly folded olfactory epithelium. So when we say here olfactory epithelium, what's down here doing the sensing is in fact this. And this olfactory epithelium is called a rosette. This is what they look like. They look kind of like a flower, the petals of a flower. These are the rosettes. And all these folds here are providing surface area, and they're covered with sensory cells that can detect compounds. So the receptor cells then send nerve impulses to the brain. The structure of the rosette and olfactory sacs is related to the olfactory sensitivity of a fish. So the more extensive the folding, the greater the surface area available for sensory cells, and therefore, the more sensitive the sense of smell is in that animal. Likewise, a lot of times tubular nostrils are also associated with a very high sensitivity to smell. So look at this, for instance. Here's an eel. This is the American eel versus a trout. Notice that the eel has a lot more folds compared to the trout. This means then that the eel has a higher sensitivity of olfaction compared to a rainbow trout. It's less derived here. And so eels actually, when we talk about them migrating out to the Sargasso Sea, they actually smell their way out there. They actually use a sense of smell to smell their way out to the Sargasso Sea. So very high degree of sensitivity here. Rainbow trout are mostly a visual predator. And so, for instance, their eyes are a lot more well-developed, but maybe the olfaction's not necessarily the best sense for them. And so they got enough here to do the job, but not nearly. If you were having a smelling competition, the eel would win that, okay? So you can look at the structure actually and can infer a lot about biology. Hagfish and lampreys have a single nares or nostril and a median olfactory sac, right? Located in the middle of the face or the head. In hagfishes, again, this is kind of an unusual thing. The nasopharyngeal duct connects to the pharynx so that hagfishes actually can smell water as it moves into the gills. And remember, hagfish have that single large nostril on their head, and that's how they breathe. Water comes in through it and then into their gill pouches. And so basically, as water's moving through that, you might as well detect what's in it. So we're going to go ahead and throw some olfactory receptors there. Lampreys have a lone medial nostril that leads to an olfactory chamber with a dead end. It does not connect to the nasopharyngeal area. So they are more similar to the rest of the fishes in the case that the nostril in the hagfish, or the lamprey rather, is a blind sac. The only difference is most fish have two, the lamprey has one. And so that's the sense there of olfaction. And again, when we look at olfactory senses, we're going to talk about taste next, really how this is relayed to the brain sort of is really what determines if it's an olfactory sense. If it's sensing smell, that is more likely going to be processed in the olfactory centers of the brain, as opposed to the taste centers, which would then process taste. And so with that, I have a video here showing the Schreckstoff reaction. Remember, this is the scary stuff. And again, Ostariophysian fishes show this. So fishes of family Cyprinidae, the minnows, carps, and suckers, and also Ictaluridae, the catfishes. They also have a very high sense of hearing or sound, as well as taste, or smell rather. And so here is the Schreckstoff reaction and how these fish sense it. And again, it's through olfaction. They're literally smelling it. And so let me play this for you. And the substance that they're going to be detecting is called an alarm substance. It's something that the fish itself produces. And so this is them pumping in some other chemical that's not this alarm substance, which they've isolated from the fish. And typically, the alarm substance is released when the fish becomes damaged. For instance, a predator bites it. Notice the fish are just sort of swimming around towards the surface of the aquarium, not doing anything special. This is the control substance, and so they don't really care. And so they're going to now put in this Schreckstoff substance. Oops. Somehow that video unadvertised. So now they're going to start pumping in this alarm substance. And you're going to see then the fish respond to it. And it's coming in up here through these little hoses. So notice that they're sort of sensing it. Now they're starting to swim a little bit erratically, like they know something's wrong. And they're going to try to get as far away from these two hoses as possible, because that tells them that something bad is happening over here. And so they're going to start swimming around. This is like a fight or flight response. They're trying to escape and get away from that. And they can sense this by smelling it. And it's usually conspecific. So if one member of this particular species is injured by a predator, that compound is released into the water column, and the rest of the school then swims away to know that bad stuff's going on. So it's kind of an adaptation. And after a certain period of time, then the water is clear of that substance and the fish go about their business. So pretty cool. And again, the sense of smell. So it's not just about finding food to eat, so to speak. It's also about other kinds of adaptations. When we talk about taste, there are 3 kinds of taste receptors. They're what we call taste buds, very similar to the taste buds that are in your mouth. You know, it's a bundle of these sensory fibers here that have these little hairs that stick out, and these things sort of then detect chemicals that come into your mouth, just like here in the fish. They're not always located, though, inside of the mouth of a fish. For instance, cyprinids, family Cyprinidae, they've got taste receptors on the outside of their lips. So the outside of their mouth, their lips can taste, not just their tongue, for instance. There are free nerve endings. And these free nerve endings, basically, this is why, again, olfaction and gustation are tough. The free nerve endings look just like the sensory fibers on the olfactory rosette that's used for smelling, but the nervous system relays it to the taste center of the brain. So it's essentially the same nerve, or it's the same sensory cell that you'd see in olfaction, but it's relayed to the taste center of the brain. And so that's kind of crazy. Sometimes they're located in the mouth, other times they're located elsewhere. For instance, they can be located on the fins, the pelvic fins of family Gattidae, the gattids, the codfishes. They actually have these taste receptors on their pelvic fins. And also the mental barbel, the chin barbel. Spindle cells are taste receptor cells similar to those in taste buds, but located on the fins and skin. So, for instance, these things resemble olfactory sensory cells, but let's say we pull one of these cells out of a taste bud and just stick it on the barbel of a catfish. Those are called spindle cells. They're basically loose taste buds. The barbels of a catfish don't have taste buds per se on them. These are usually relegated to the mouth or like the lips, but they might have spindle cells which essentially function the same way. And so as the catfish then, family Ictaluridae, swims along the bottom, it can then taste everything below it in the detritus. So kind of cool. They can taste all around their body, not just what's in their mouth, what they're swimming over, what they're touching, things like that. Actually, before it even enters their mouth, they can taste it. And again, it looks very similar to the olfactory receptors in some cases. It's just a different part of the brain that processes it. When we look at hearing and balance, they're sort of tied together, and so we look at hearing and balance, they're closely related. Anyone who's done fisheries work knows that there are these otoliths in a fish head. You know, for instance, this largest of the otoliths is the sacculus. It's located in the saccula or the saccule. And what you can do is you can kill a fish and cut it open and crack its skull open, and you can actually pull this otolith out of it, and you can cut it in half. And there are little rings in there. You can count the rings of the otolith to tell you how many years old that fish is. So you can age a fish by sectioning that otolith, and it's usually the biggest one, the sacculus. And so what these stones do is they're literally stones or very dense bone, and they're located inside of this labyrinth organ, also called these semicircular canals. And what these are is these are dense. And so they function in hearing because if a sound wave travels through the water, and again, going from the density of, say, water to the density of bone, again, we've sort of, we've changed the density of the media in which the sound wave is moving through. We've gone from less dense water to more dense bone or stone, which means then that this stone will vibrate, and the fish can hear that. Also, as the fish moves, these stones can move. They're sort of embedded in a gelatinous matrix. And so this then allows the fish the sensation of movement. So it can then move in 3 dimensions: up, down, left, right, back, forward. Also X, Y, Z from geometry, your 3 planes. So they know their orientation of movement, as well as then they can vibrate in the presence of sound. So that's why they're 2 tied together. The stones are more dense than water, so they vibrate to sound waves to aid in hearing. And in the case of most bony fishes, the teleosts or osteichthyes and the elasmobranchs of chondrichthyes, there are 3 semicircular canals. They can see or they can feel or move 3 dimensions. However, the lampreys have only 2 semicircular canals, so they can only see in 2 planes, X and Y. They cannot see the third. They cannot feel the 3rd axis of movement. And hagfish, they only have 1. They literally have a single pathetic semicircular canal. And I say pathetic. What do these things do all day long? They just tumble around on the bottom of the ocean in the muck, in the darkness. They don't need eyes because it's dark. There's no light down there anyhow. And all they do is tumble around. They need to have 1 semicircular canal. And all they need to know is, are we moving? Are we tumbling? If the answer is yes, all right, keep tumbling. If the answer is no, then maybe we should start tumbling, or the opposite. You know, we've been resting a while, let's start tumbling again. So they just have one semicircular canal just to see if they're moving around. So they don't really need to have sophisticated swimming. They've got other things. It's not like they're swimming away from a predator, because they just produce a bunch of mucus every time something tries to bite them. And so that's what they do. So these are then the hearing and sense of balance. Very similar to other vertebrates such as yourself. Our ear has 3 semicircular canals, and we also have ear stones. Very similar. When we look at fishes of Ostariophysi, which again, the minnows, which is family Cyprinidae, Catastomidae, and Ictaluridae, including the other silurids, we have this Weberian apparatus. And again, when sound waves are traveling through something as dense as water, it wants to go into some other media, whether that's either more dense, like a stone, or less dense, like gas. When the sound wave transitions into that different media, that's when we can sense it. So what fishes of Ostariophysi do is they have this swim bladder filled with gas, and the sound wave goes through the water into the body of the fish, which is mostly water, but then boom, it encounters something less dense. Well, this then vibrates and amplifies a signal, which is then passed from gas to bone, which is more dense. Those bones then transmit this signal to the inner ear, where it is then perceived at a very high efficiency. So these fish hear very well. The bones of interest here are the tripus, the intercalarium, the scaphium, and the claustrum. And so these are the bones then that relay that sound wave, or propagate that signal of sound waves to the inner ear. And again, these are just bones of the skull to show that the inner ear, this labyrinth organ, if you will, is located on the inside of the ear. And that's exactly what it's doing, is it's sending this vibration up into the inner ear, where then it can help to vibrate these stones to help with that sense of hearing. Another way fishes hear, again, is by touching sound, or hearing touch, however you want to look at it. The lateral line, the acoustico-lateralis. This system contains pores with fluid-filled canals that connect to neuromast cells that transmit information to the brain. So, for instance, this lateral line goes down the side of a shark, or down the side of this perciform. This looks like a largemouth bass. Again, we can tell by the dorsal fins, right? And so this allows fish to feel, feel, however you want to say it, near field vibrations of sound within the water. So even if murky or dark water, no light, fish can still determine the location, speed, direction, movement, and size of objects moving towards them with their lateral line. They don't need to see it, they can just feel it. And they don't, when we hear a noise, we sort of say, oh, we heard something, but we can't like sort of feel, we can sort of indicate that was loud or that wasn't that loud, but they can actually like feel how fast that thing is moving towards them as well. So similar to how you might feel like when a big truck drives by, you're standing on like the side of a road and a big truck drives by with a swoosh before it gets to you. Imagine if you could feel that sensation 20 feet away, like when you saw the truck, a long distance down the road, and you're like, I can feel that truck coming at me. Now, because I said fishes have a blind spot in front of their head because of their lateral placement of the eyes, notice that these canals of the lateral line are located all on the head as well. And that is because this lateral line is what helps them, because they can do location and direction of movement. This allows them then to overcome that blind spot. So the lateral line of a lot of fishes is very sophisticated on the head. And in some cases, like the moray eels, they don't even have to have this down the side of the body lateral line. Their lateral line is almost always relegated to their head only. So the way the lateral line works is like this. There is a series of canals down the side here or in the head. And each of these canals is literally, you can see these little spots down the side of the fish. They're pores, they're open pores. And what happens is water comes down towards the fish and then goes into one of these pores. And it goes into a series of canals located, embedded underneath the scales of the fish. And then when water passes through these canals, they pass by these things called neuromast cells, these sensory cells. Here's one right here. And the top of this neuromast has a cupula. And a cupula is like a big gelatinous mass. It has all these little like hair-like fibers embedded in it. What happens is as the water passes through here, it bends this cupula one direction or the other. Like it bends it to the left or bends it to the right. And what happens is when that thing bends, these sensory cells down here receive that information and relay it again to the brain. And so, like, for instance, how fast the water molecule's moving, how hard it's moving, what direction it's moving. You know, like, for instance, is the object approaching from the back of the fish, bending these things this way, or is it moving towards the front of the fish, bending it this way? They can then use this information to get information on direction, location, speed, and also size of the disturbance. What's disturbing the water coming towards us? And these structures here, these neuromasts, do this. And so I like to think of it like this. You know, we started talking about black holes and whatnot with the Nobel Prize. You know, people say there's no sound in space, which is kind of wrong. There is. All sound is one particle striking another and transmitting a vibration. The reason why there's no sound in space is that space is 99.99999 something percent a vacuum, the absence of everything. And so if one molecule were to be excited or pushed in one direction, it's not like it's got anything to bounce off of. That's what creates sound. So when you hear sound, it's because, you know, you've got billions of molecules of nitrogen or oxygen or whatever floating around in the atmosphere. And as you speak, you're creating a disturbance that then is all these gas molecules bouncing off of each other. In the case of water, it's the same thing. Water molecules are bouncing off of each other. And when one water molecule bounces, it sends that to the next one, and it sort of propagates this wave. And so this is why it's acoustical lateralis, because again, if one water molecule is bouncing off of another, and this propagation of this wave continues to propagate towards the fish, in here, bending that. This thing functions then very similar to those vibrating stones inside the inner ear, at least as far as perceiving the sense of what we call sound, which is really just a series of particle collisions. In this case, the water molecules outright all along the side of the body of the fish. And because we're sort of not necessarily measuring it by vibration, we're measuring it by, as these things pass over these fibers, it can bend it in one direction and the fish can sense that. So here's a video that sort of explains this. And it's pretty cool. Again, Smithsonian, hopefully they don't give us another commercial here, but the shark's entire body is an ear.
In a rare and unique experiment, shark expert Dr. Cliff and sensory biologist Dr. O'Connell will dissect this great white.
Okay, so just here behind the eyes are two pores. Let's see if we can find them. These are the external openings to the ears.
You've got to look very carefully. The openings are very, very small, so there's no external ear at all. It's just literally a pinprick opening reaching down inside.
Is just below these holes is a fluid-filled inner ear.
Nearly invisible to the naked eye, two tiny openings on the surface lead into the shark's internal ears, embedded deep within the skull. The shark's inner ear is filled with fluid, giving it direct contact to its ocean environment. While three D-shaped tubes are responsible for the shark's balance, orientation, and body positioning in the water, similar to the function in a human ear.
Now this shark's auditory system allows him to detect sounds that we can't hear, very low frequency sounds, such as those associated with the wounded fish.
So that struggling fish is just like you and I recognizing the dinner gong. That
Absolutely.
A struggling seal creates an irregular pattern of splashes, and it's these sounds that attract the great white shark. The sound waves travel down into the shark's inner ear. Within this chamber, thousands of minute sensory hair cells called cilia line the walls. As the incoming sound passes over, it causes the hairs to bend. This movement triggers the release of an electrical signal directly to the brain. The great white may use its ears to listen to the seals, but it has another predatory power so advanced it can hear their movement from all around its body. The lateral line, a sense that will lead the great white directly to its prey. Buried just below the surface of the skin, a narrow tube runs either side of the shark's body. It's known as the lateral line.
there's that canal.
That's incredible.
This is the lateral line, a small, hollow tube which acts like an extension of the ear. The shark can hear all along its body. The pressure waves produced by the seal's movements pass through tiny pores on the skin surface and down into the lateral line. This main canal is lined with thousands of minute sensory hair cells, similar to those found in the shark's inner ear. These hairs are so sensitive, they can detect the slightest vibrations and turbulence in the water from hundreds of feet away. As soon as the hairs move, they stimulate nerve cells, which fire a warning signal to the brain. This early warning detection system encircles the shark's entire body, which means it can receive a constant and continuous update of the seal's location. Thanks to the inner ear and the lateral line working together, the great white can calculate the size, distance, and exact direction of the seal.
Sitting down.
All right. So that is the acoustical lateralis. Very sophisticated. Again, combining the senses of hearing and touch, very interesting. And again, as water molecules come into this canal, it displaces these neuromast cells. Another interesting thing that fishes have here is what we call electrical reception. Now, not all fishes have this. There are what we call ampullary receptors that are widespread among fishes, and they've been identified in lampreys, for instance, Petromyzontiformes, sharks, chondrichthyans, the lungfishes, Ceratodontiformes, the African reed fishes, Polypteriformes, which are the fakirs, the coelacanths of Latimeridae, sturgeons in Chondrostii. Also, that's Acipenseridae is the family, but also Polyodontidae, the paddlefish. The bill of the paddlefish has ampullary receptors in it. Several orders of more advanced bony fishes, including Osteoglossiformes, Gymnotiformes, and Siluriformes. So these are the catfishes. So when we talked about the electric eel, and I said the electric eel is really not an eel, it's more closely related to a catfish. The electric eel is another example of electrical reception. And of course, we talked then about chondrichthyan fishes having ampullary receptors, but also then things such as the electric ray in family Torpinidae. These receptors typically are located in small pockets in the skin and at the end of a canal is filled with some kind of electrical conductive jelly. Primarily made up of mucopolysaccharides, which is just a way of saying a mixture of proteins and carbohydrates, and salt that allow electricity to be conducted very well from the water through this jelly to then some cells which can sense it. And again, the nervous system then sends that information to the brain. So ampullary receptors are responsible for passive electroreception. So we're not talking about like echolocation per se, where the fish sends an electrical signal out and then sees if it receives information back. This is more of another fish or organism is creating electrical current, and the animal with this sense then can detect that electrical current. So ampullary receptors respond to low frequency stimuli, including those that are the result of physical environment and those of biological origin. So for instance, a muscle contraction results from a shift in ion polarity of cells inside of an organism, which is a miniature electrical reaction. A fish with these senses can feel that, right? It's something as much as a muscle twitch. Also the physical environment. Anyone, it's chemistry. A water molecule, if we recall, one oxygen, two hydrogen atoms held together, and there's a dipole moment, right? Where oxygen is electronegative. It likes to pull all the electron clouds close to it, and that leaves then the hydrogens without their electrons. And so that means that each of those hydrogens are a little bit more positively charged. The oxygen side is a little bit more negatively charged. And what happens then is as water molecules flow by themselves, those positive and negative forces sort of interact. And depending on the rate at which they flow or the pressure at which they flow, there's actually a little electrical static discharges as each water molecule bounces by another. That creates a little electrical current. So a fish, for instance, can sense flow of water, hydrogen shearing, things like that. So abiotic and biotic sources of electricity can be sensed using these ampullary receptors. So the ampullae of Lorenzini are probably the most famous, and these are these little pits, these little dots here. These are the openings of these little pores filled with conductive jelly. So if you look inside of each one of these little pores, there's going to be filled with conductive jelly, and then there's going to be some sensory cells in there. So the electric current comes in, transmitted through, sensed, send that signal then to the brain. So they're unique. Ampullae of Lorenzini are unique to elasmobranchs. They can detect within the centimeter close range, and a system of pits concentrated in the head of sharks oftentimes. Again, this sort of helps them with their fact that they've probably got a blind spot in front of their head. They can detect weak electrical fields created every time the muscle contracts in a prey item, for instance. A flounder buried under the sand, things like that. Sensitivity is 10 times greater than the output from the prey. So these things are very sensitive when it comes to being able to detect these things. And the jelly-filled canals that lead to cells then that transmit information then to the brain. And so they can detect buried prey, stuff that's hidden. They don't have to use their eyes to see in low light. They can actually just feel if something's around them because of the electric currents that those things produce. And so with that, I'll show a video here of the ampullae of Lorenzini. This is a pretty cool video because it shows some stingrays honing in on electrical impulses. You can sort of see the behavior of the fish.
We live in a world full of electric fields. Animals, plants, even the earth itself creates them, but only a handful of creatures can actually sense them. Sharks, also rays and skates, a whole group, animals called elasmobranchs, can actually detect electricity. Maybe it's like feeling the presence of someone hiding in a dark room. A different way of experiencing their world. An extra sense that we don't have. And for sharks and rays, an extra edge for finding their prey. The discovery of this exceptional sense dates back 300 years, long before the discovery of electricity, to a man named Stefano Lorenzini. Lorenzini was a doctor with a passion for anatomy who one day found himself dissecting a ray, which is where he noticed the strangest thing. Tiny pores that dotted the ray's skin. He drew a picture. It shows how each pore led to a tube full of goo, and each tube ended in a little pouch. Those little round pouches reminded him of ancient Roman flasks called ampullae, so that's what he called them. But when it came to the function of those ampullae, Lorenzini was stumped. 3 centuries later, we're still figuring out exactly how electroreception works. And a big part of that is studying what makes these electric fields in the first place. Every time this fish opens its mouth to breathe, it exposes its mucous membranes to the salty water, creating a tiny voltage that disappears every time the mouth closes. At Florida Atlantic University, scientists are actually able to measure this phenomenon. That repeating on-off pattern is an electric frequency carried easily through salt water. 2 hertz, 2 breaths a second. Stingrays like these can tune into that same frequency, like a radio channel. Take a close look at a stingray and you can see those pores. They lead to long canals that cluster together. When an electric charge enters a pore, it travels through the highly conductive gel in the canal to the ampulla, where special cells detect the current and send a message along nerves to the brain. That's how stingrays can spot prey hidden beneath them, even though their eyes are on top of their heads. But the ampullae aren't just for hunting. Pores on the ray's back also let it know if someone's trying to sneak up on it from behind. Here, the researchers placed diodes on the floor of a tank, replicating the same electric frequency as a breathing fish. They flip the current from circle to circle, and the ray strikes at the active target. Maybe by studying how this sense works, we can learn better ways to coexist with these ancient creatures. Find new ways to keep them out of our nets and away from our surfers. Of course, none of this really explains what it's like to feel electricity. Is it as simple as flipping a switch on and off? Or as varied and subtle as smell or taste? A whole spectrum of experiences hidden from the rest of us.
So that's the ampullae of Lorenzini. You can see it's pretty cool. I always like that part of that video where they show the stingrays honing in on the spots in the tank that they charge. It's pretty cool. We look at other types of fishes. You know, again, the ampullae of Lorenzini are in the chondrichthyan fishes, but when we look at other fishes, we have what are called tuberous receptors. And these are responsible for active electrolocation. So the detection of an electrical field produced by the fish's own electrical organs. And so we talked about the African, or really the South American electric eel that can actually discharge electric currents. And then these fishes can then look for or sense a disturbance in that electrical field that they generated. And they process that information. So this is more like electrolocation. They create an electrical field, and then something swims into it, disrupting it, and the fish can sense that. So these are only found in those fishes that can generate electrical discharges, or EODs, such as the gymnarchids and the catfishes of Africa and South America. And so, again, the electric eel. And so electroreception and muscle cells have been modified in species. We talked about this with the electric ray, and that oftentimes muscle is used to create electric currents, and then these electric currents are then used, for discharge. And so it's usually modified muscle cells that form electrical plaques or electroplaques, they create a discharge, and then a disturbance to that discharge can then be perceived by the same fish creating the discharge. So pretty cool. So here's a video now on the electric eels from National Geographic.
In murky tributaries of the Amazon River, one of South America's apex predators uses what could be the strangest hunting tactic of all. This 6-foot-long animal produces what almost no other animal can conjure: a killing shock of electricity. Known as the electric eel, it's a unique creature, more closely related to a catfish than an eel. First, it produces a small electric field, sensing distortions in it to navigate and to find other animals. These fish have no idea that their heartbeats and muscle action are drawing a killer closer. The electric eel probes likely hiding spots. The eel is not going to rest until it's eaten. Sensing prey, the eel flushes it out into the open and delivers its death blow. 500 volts, enough to stun an adult human. Quiet until hunger moves the eel to hunt again. Full, the eel disappears into the murk.
So that's a slightly different take on electrical reception, which is these ones here are passive. This one here is more active. They create an electrical field and then sense disruption to it. Now, I said that there's potentially an eighth sense, and that eighth sense here could be related to the electrical reception, which is why I stuck it here: the detection of electromagnetic disturbances. So some fishes, such as elasmobranchs, tunas, eels, salmonids, have the ability to detect weak electrical fields that they create as they move through the Earth's magnetic field. So we're talking about latitudinal or longitudinal movements. So this provides fishes with the indirect way then of sensing the Earth's magnetic field, and it gives them some directional information with respect to the compass. So for instance, are they swimming north, south, east, or west? And so fishes, as they move through the electrical field of the Earth, somehow are able to understand compass direction. And this is important during, like, for instance, homing migrations of salmon. As the salmon are returning, they have to know, hey, if we're swimming across the Pacific, you got to swim towards North America, not towards Asia, right? Somehow they understand this. And the mechanism for this sensing of magnetic fields is unknown. We actually don't know how fishes do this, but it would be important then for migrations. We just know that fish are able to do this. And it's lack of the other senses too. Like I said, eels a lot of times will smell their way to the Sargasso Sea and things like that. They've plugged the nostrils of salmon and released them, and the things return to their natal streams. They've done all sorts of stuff. We don't know how they sense this, but our guess is it's probably related to either ampullae or tuberous receptors, some way to sense electrical and or magnetic fields. So that's another really new sense that we're still trying to understand. But fishes obviously have the way to do this. So with that, I have a story. Now, this is you learned it here at AEC 441. Can fish read? Well, of course not, right? Well, actually, we don't know. So fishes have been trained to recognize different shapes, including closed shapes like squares, triangles, and circles. Open shapes, for instance, shapes that are not enclosed, and even letters and words. And they can recognize this because they can be trained. If fish are provided with a food or a reward as they swim by a selected shape, they can be trained then to know that if every time you swim by the letter C, we give you food. They know then to hang around the letter C, and they can tell the difference between the letter C versus the letters A and B. And so a fish then can choose the image that's associated with the reward. And they can actually do this pretty well. So although fish might not be able to write a best-selling novel, they demonstrate remarkable memory. So the archer fish here, shown here, is a tropical fish that literally at the surface, it comes to the surface and then shoots a jet of water out of the water. Now, in its natural environment, what these things are doing is there's insects, for instance, in branches or leaves above the surface. The archer fish can see the insect there, and it intentionally shoots a stream of water, knock the insect into the water, and then they go and eat it. That's why they've evolved this. So what they can do, though, is scientists have trained fish to shoot a jet of water at an image that they get rewarded by. So if fish are presented with two human faces, pictures, and they're trained to choose one of them using a water jet by squirting the face, the fish can do this with about 80% accuracy. So they say, hey, this face here, if you shoot a stream of water at it, you get a food pellet. If you shoot a stream of water at this one, we're not going to give you anything. And they understand which person to shoot the stream of water at. And they can actually do this about 80% accuracy. And they can actually even do this when more obvious features, such as they can remove eyeglasses, or they can take away the hair, and it's just the person's face. They can actually eliminate certain features of that person, and they can still see this and still recognize it, or a black and white image versus a color image. So if you want to read more about this, it was published in Nature Scientific Reports here. This is actually a pretty cool article. It's one of those, I always like to see when I see people doing studies like this with fish. It's very interesting. So with that, I have one very short video on the archer fish, and then I will entertain questions.
Think of the archer fish as a fastball pitcher, except that the distance to home plate changes with each batter. Plus, a baseball holds its shape, and the archer fish has to shoot a jet of water. But researchers have found that the archer fish can control and shape that jet in a way that no other creature does. To study this technique, two researchers from the University of Bayreuth in Germany first spent about a year training archer fish. The fish had to shoot water from just the right spot in the aquarium so high-speed cameras could catch the action. What the scientists found was that whether the target was at about 8 inches, or 16 inches, or even 24 inches, the drop that slammed the target, that bullet at the end of the jet, came together for maximum impact at just the right moment. So how does the fish do it? It doesn't just spit harder. It opens and closes its mouth in a way that varies the speed of different parts of the water jet. Quite a trick. And humans use water jets for all kinds of purposes. This new understanding of the archerfish technique could lead to improvements in everything from surgical tools to fire hoses.
Pretty interesting. And so also that shows you, I talked about how vision is important because the refractive index of water is different from that of air. The archerfish obviously also has an adaptation so that it can see outside of the water surface into the air very well as well. Also, so that indicates then that not only are they very good at shooting those jets of water, they've got very good vision both in the water and outside at the interface, where the water meets the air. So with that, I'm going to stop recording